Hi everyone, welcome to this week's Single Cell and Spatial Exploration Club. My name is Julie. I'm a scientist here at BioTuring, and this week we'll cover clonotype analysis, which we just released on the BBrowserX Pro earlier last week. So let's go over the agenda for today. So there are two things that I want to introduce here. The first is clonotype analysis, but the second we also released with clonotype analysis. This is our new interface for BBrowserX Pro. So you notice in the last couple of weeks, we released the new interface, which now looks a lot more like SpatialX and SmartBulk. So some of the new highlights for this new UI include us wanting to maximize the screen space for the TCR and UMAP. We also consolidated all of the analytical tools in one single toolbar on the left-hand side. And then we also transitioned from, instead of using pop-up modals, we now use the slide out panes so that it's kind of less annoying to use the tools and have them cover the screen and everything. And then finally, we also unified the gene query panel and the metadata panels on the right-hand side as well. So in our documentation page, I also noted the changes between the old design and the new design here, so you guys can check that out. And next, to highlight the clonotype feature here. So essentially, this tool will help you analyze your T cell receptor and your B cell receptor data. This type of data is really useful in finding out how your cells are responding to some sort of antigen in your immune system, right? And in particular, the region that we are interested in on the T cell or B cell receptor is called the CDR3 region. This region is the most variable region on the receptor. And so it's really important within this type of analysis to figure out what CDR3 regions are important for us to know. Okay, so again, the CDR3 region is the most variable, and it is the region which actually comes into contact with the antigen. And so when we study TCRs and BCRs and how they come into contact with an antigen, these T cells and B cells will typically undergo clonal expansion, meaning the same cells with a particular receptor sequence will expand and have the same receptor sequence on its cell surface. And so this clonal expansion is an indicator that some positive immune response is occurring within the body. And so analyzing TCR and BCR sequencing data is pretty useful in, for example, vaccine development or in, other types of disease type of research. Okay, and so some of the visualizations that are output from this tool include this stacked bar plot here. This is where you can find which clonotypes are expanding. So you can see across your different samples or across your different subjects, you'll be able to find the expanding clonotypes. Other than that, you can also find which clonotypes are shared between different subjects or samples as well. So this is particularly useful if you have treatment data. You're comparing before treatment and after treatment, and you want to see, okay, are these patients responding after the treatment in the same way? And one way you can evaluate that is by finding the shared clonotypes between those patients. Okay, next here, if you want to find where this clonotype tool is located, you'll actually have to enable the new layout on BBrowserX Pro. So within this month, the new layout is still going to be in the beta period. That's because we're still collecting feedback to see what users think about this new layout. So you'll be able to toggle back and forth between the old and the new, and then we'll be happy to hear any feedback that you guys have. But again, the clonotype analysis tool is only going to be available on the new layout. For the data set that I'm going to be going through this week, you can find the data set on Geo here if you search the GSE ID or the title here. So one kind of unique thing that you have to do is that we do have a lot of readily available TCR type of data sets on Talk to Data. But what you'll have to do is you'll have to request our data team to upload these TCR sequencing files for you so that you can access the clonotype tool and run it however you want. So in the case that you want to upload this data set yourself, you can go ahead and download these necessary files from Geo. Okay, next let's dive into the experimental design. So the question that I want to tackle here in today's webinar, it has to do with this inflammatory arthritis disease. So in this particular data set, the authors have taken samples from patients with rheumatoid arthritis and psoriatic arthritis, and they sampled the tissue from different tissues. So they have blood samples, synovial fluid samples, and synovial tissue samples. And one of their main questions they want to ask is, okay, can synovial fluid actually serve as a proxy for investigating synovial tissue T-cell infiltrate in inflammatory arthritis? So this is kind of more of a non-invasive way of trying to see if these patients with inflammatory arthritis are experiencing some type of T-cell infiltrate. And instead of taking the tissue sample, we can more non-invasively take a fluid sample instead, and then do some research that way. Okay. All right, so now I'll show you guys how you can actually run the clonotype analysis tool here on the BBrowserX Pro. So let's switch over to the app here. Down here, you can find our clonotype tool. We'll open it up, and there's three main steps that you have to fill out. The first is you have to specify which receptor type or sequencing data you have, either TCR or BCR. The next one is where you can upload your file. So for the file types that we currently support on BBrowserX Pro, we have support for 10x genomics and parse bioscience. In the next coming weeks, we are also planning to support BD Rhapsody as well. So you guys can stay tuned for that. But in particular, for this data set, the authors used 10x genomics. So I uploaded the TSV file here, or the CSV file. So for those of you with 10x genomics here, typically the file type is going to be named filtered_contig_annotations.csv. And this is kind of a screenshot of what the contents of that folder will look like. But usually you should have the cell barcode within that file, as well as the contig ID, and some other columns such as the VDJ genes and the CDR3 sequence as well. Okay. So after you upload this file here, next is the parameters. So the parameters here, you're basically telling the tool how you want to define a clonotype. So there's a lot of different ways in which you can define a clonotype. So you can compare the receptor sequence by either its amino acid sequence or its nucleotide sequence. And then furthermore from that, you can also specify the clonotype cluster based on the VJ genes or the VDJ genes. So let me elaborate a little bit more on what that actually means. So how do you actually define what a clonotype is? For us, we can divide the definition of a clonotype into two main categories. The first category is you can compare the CDR3 nucleotide sequences. The second category, you can compare the CDR3 amino acid sequences. So I think it's pretty standard that a lot of people will want to compare the amino acid sequences instead of the nucleotide sequences because, as you know, a lot of nucleotides can kind of map to the same types of amino acid sequences. So for example, if I were to choose to compare by the amino acid sequences, then I can further break that down into whether I want to compare the alpha and the beta chains across T cells. If I only want to compare the alpha chains across T cells, the beta chains only across T cells, or have any of the criteria be met above. So typically, when I analyze TCR sequencing data, I will choose this fifth option here, where the alpha chains and the beta chains of a T cell must match in order to be considered the same clonotype. Because, typically if a T cell receptor has the same alpha and beta chains, then that makes it a functional T cell receptor, right? Okay, and so let's dive into the report here. Let's open up this report. So as I explained earlier, there's two main visualizations that are output from this tool. The first one here is the expansion. So this is where you can see which clonotypes are actually expanding. And how you can find the definition of what an expanding clonotype is you can actually configure a range here. So what we're looking at is the clonotype sizes kind of categorized into 4 different categories here. So clonotypes with at least 1 cell are categorized as blue, versus clonotypes containing at least 5 cells are categorized as red. In my definition of an expanding clonotype, let's say I want to see clonotypes with at least 10 cells or more. So this could be pretty subjective or pretty trivial, but I see for my small data set here of only 36,000 cells, even with a threshold of 10, I can still see some expanding clonotypes here. So now what I'm actually interested in is what cell types the expansion is occurring across. So I have the author's cell subtype here. And from what I see here, I see a large population of expanding clonotypes occurring within my CD8 positive, GZMK positive T cells. Now, when I looked further into what type of T cells these are actually a type of or a subtype of cytotoxic T cells, but instead of promoting cytotoxicity, they actually promote inflammation. And so that's what I've summarized here in these slides. So of course, we're looking at samples from patients with inflammatory arthritis. And so we expect to see a large population of T cells that are pro-inflammatory. And so we could observe that within this expanding clonotypes here. Furthermore, what you can do is you can actually click on the bar here, and that will expand the data table down here. And so within the data table, you can sort it by the clonotype size. And now what you'll be able to see is all of these expanding clonotypes, as well as their CDR3 amino acid sequence and their V, D, and J genes. So you can see here how these 2 rows are assigned to the same clone ID. That's because this first row here is corresponding to the alpha chain, and the second row is corresponding to the beta chain. And so that's how you can interpret some of the results and data tables that come out of this tool. All right, so let's move on to the second visualization that's kind of output from here. So the second one here, you can move over to the shared clonotypes. And again, this is for you to see what clonotypes are similar across, for example, across your different T cell populations or across different samples. In my particular results page here, I see that there's a large number of shared clonotypes between my CD8 positive inflammatory T cells and my CD8 positive memory T cells. So that's interesting to see that when these inflammatory T cells are kind of expanding in their clonotype, this might be also telling me that these clonotypes are now becoming memory T cells and are circulating within the immune system to kind of protect against inflammation. And so that's kind of the conclusion that I drew from this shared clonotype heat map here. Again, I have the conclusion here that I think maybe these memory T cells are being populated with the same clonotypes as the pro-inflammatory type of T cells. But let's go back to our original question, the main question that I had. The main question that I had in the introductory slide, which is, can synovial fluid serve as a proxy for studying synovial tissue? And so now when I looked at the shared clonotypes across the different tissues that the authors have sampled from, I see a large number of shared clonotypes between the fluid and the tissue here. So this is really encouraging to see that, yes, possibly we can use synovial fluid to study inflammatory arthritis instead of synovial tissue, right? And again, if you click on the node here within the heat map, it will also show the same type of data table at the bottom, where you can see the CDR3 amino acid sequences as well. Okay, so to demonstrate that really quickly, let's go and change this to the tissue, for example. Okay, and then I can click on this, and then it will show you the data table here. So from here, you can also save some of these CDR3 amino acid sequences and input them into some online database to see if they match with any type of antigen or epitope that's within that particular database. So that's one way you can kind of further perform some analysis on this data set. Okay, and so that is the end for how you can use the clonotype analysis tool here on BBrowserX Pro. If you want to learn more about the analytical tool, we also have our documentation page, which also dives into the methods of how we perform the data filtering on our tool, and then also how you can define what a unique clonotype is. And it also shows you how to interpret the results again. Okay, so in a second here, I'm going to open up the poll because this is the first iteration of our clonotype analysis tool. And so in our next iteration, what we want to know is what you guys are interested in seeing next. So some of the things that we might add onto our tool is, for example, diversity analysis. That'll tell you how diverse your clonotypes are within your patients. Antigen or epitope prediction. So once you have your CDR3 sequence, what kind of sequences will that receptor kind of recognize? Or other things such as, um, you know, sorry, I can't remember off the top of my head right now, but those are the two main things that we are looking to add to our tool in the next iteration. Okay. And then this slide, of course, for each SCSE club that we have, we also re-uploaded to our YouTube channel, so you can check out our playlist on YouTube if you want to rewatch as well. So we're opening up the floor to any questions that anyone has. And of course, we also appreciate any feedback you guys have as well. Okay, I think I'll give it a couple more seconds for people to answer the poll. But in any case, this is the end for today's SCSE Club. So thank you everyone for joining this week, and we hope to see you in the next 2 weeks. Hey, thank you everyone.